Hello, and welcome to our CBE205 group project. Today, we'll be talking about metabolic engineering. So, what exactly is metabolic engineering? Metabolic engineering is a discipline of engineering that combines the technology of genetic engineering with the breakdown analysis of an organism's functions as it's often performed in systems biology. Using these techniques, metabolic engineers can observe a cell's metabolic pathways, which are networks of biochemical reactions, to further design modifications that specifically improve its biological functions for the purpose of influencing them to produce useful chemicals and products. This is the guiding principle behind metabolic engineering, envisioning cells as biological factories that can always be improved upon with careful planning. So then, when a metabolic engineer sets out to do work on an organism, what do they aim to accomplish? The goals of the process are to first study a cell's activity to get an understanding of what precisely can be altered within the cell, such as a gene producing a particular enzyme or a reaction producing a useful chemical. Once engineers know what to aim for, they can use recombinant DNA technology, among other genetic engineering techniques, to attempt to improve an aspect of the cell's activity for increased yield, efficiency, production speed, and numerous other factors. Further, metabolic engineers analyze what they learn and their results in order to continually iterate upon their target organism, striving to ensure they are economically, environmentally, and commercially viable as well as competitive.
There are three main steps in metabolic engineering. The first is the study of the organism's metabolic path. This can be done through a radiotracer technique where they place a radioactive particle in the organism and track it through the organism and its reactions. Next, a pathway that will maximize the production of the desired product will be designed. This can be done using a computational approach called knock that identifies the gene deletions that will maximize the product. Finally, the designed pathway will be applied to the organism, typically on an experimental level at first, using genetic engineering approaches. Some techniques used to maximize the production of the metabolic pathways are heterologous expression of gene clusters. This is implemented when a pathway in one organism can't be manipulated, but if moved to another organism, it can be manipulated to produce more metabolites. Another is modifying regulatory networks by changing activators or repressors, and new pathways can be made or altered by gene insertion or deletion. Some other techniques are stimulation by precursor, genetic knockout of Loci, and quorum sensing.
In the effort to reduce CO2 emissions from fossil fuels, yeast is being researched as a natural producer of biofuel. Why use yeast? It can produce ethanol and CO2 with simple raw materials such as sugar and water. Baker's yeast has been successful in the beer and wine industry because it can grow anaerobically in acidic conditions. However, the adjustments of the cell are needed for the bioethanol industry. In order to metabolically engineer yeast, we have to look into the general process of the cell. Yeast has a simple process in that sugar and water are only needed to activate the enzymes that produce ethanol, CO2, and more yeast cells. However, the bioethanol industry needs new strains of yeast that can withstand higher temperature, higher ethanol concentration, and be more efficient in its metabolic pathways. In the steps of engineering yeast, there is a key step called pretreatment. This is where yeast cells are metabolically engineered to produce at industrial settings. One example of pretreatment that can be applied to yeast is the heterologous expression of gene cluster, which is a process of inserting genes from one organism into a host. For yeast, this creates a new metabolic pathway that will help produce ethanol at higher concentrations. One example is a co-expression of two cellulosic enzymes. Here, the efficient cellulose enzymes EGI and beta-glucosidase and S-fibulagera are integrated to the yeast DNA. As enzymes are transcribed, a new pathway is created that will break down cellulose into useful sugars for producing ethanol. The success of this was the co-expression of the enzymes were able to convert amorphous cellulose into ethanol in one step, overall making it metabolically efficient to produce ethanol at higher concentrations.
Another pretreatment example is redirecting or creating new metabolic pathways. Each protein within a cell may exert varying degrees of control over a metabolic pathway and can be modified to improve metabolic fluxes. This is done through gene modification, which makes changes to a living thing's genetic information that would otherwise not occur by natural mating or reproduction. Primary targets for gene modification include DNA or gene content through adjustment of homologous or heterologous genes, transcription efficiency by using promoters of appropriate length, and mRNA abundance and translation. Yeast commonly uses gene copy number and transcription efficiency that can make the yeast product a more useful and successful bioethanol ingredient. The first example is deletion of genes such as MIG1, MIG2, or GAL80, which alleviates glucose repression in yeast. Glucose is an important energy source for yeast growth, which creates a more active and faster producing yeast product. The second example is deletion of the PDC2 to reduce pyruvate decarboxylase activity. Limiting this activity allows for larger yeast or sample growth by limiting the possibility of alcoholic fermentation.